This segment is of the circulatory system for Anatomy 2203. The circulatory system consists of the heart, a series of vessels that lead blood away from the heart under greater pressure known as the arteries. Then exchange takes place through very small vessels known as capillaries, and then a series of veins return the blood back to the heart in this circulatory manner. So it's heart, arteries, capillaries, veins in a circuit. Look at slide number 29 for cardiac muscle. Cardiac muscle makes up the myocardium of the heart, and is that type of muscle that enables the heart to have a pump-like action. And compare this type of striated muscle to that of skeletal muscles making up the limbs. In the field of view, one can see an individual cardiac myocyte or muscle cell, or sometimes referred to as a cardiac fiber. Nonetheless, it is a cell. These cells, unlike the skeletal muscle cells, are much smaller. They have a centrally positioned nucleus. In some instances, two nuclei may be present. So they're considerably smaller than the skeletal muscle cell and usually have a single centrally placed nucleus. Now, the tip of the pointer is illustrating a single cardiac muscle cell. This is the length of this individual cell, where the tip of the pointer is now illustrating to about this location. Its width extends from about here to here. So this is a single cardiac muscle cell. Here lies the centrally positioned nucleus. Oftentimes a pool of glycogen can be seen on either pole of the nucleus as shown here. Note also that individual myofibrils can also be seen at the exact tip of the pointer, giving it this sort of banded or striated appearance. Close observation, you can actually see some striations on some of the adjacent myofibrils. So this stranded appearance are myofibrils scattered throughout the sarcoplasm of this muscle cell. Note too that the muscle cell, the cardiac muscle cell, shows branching, and it's united with its neighbors by attachment points known as intercalated discs. So this is an intercalated disc as shown at the tip of the arrow, and it marks the end of this particular cardiac muscle cell. On the other end, one can see an intercalated disc here as well, uniting this particular cardiac muscle cell with its neighbor here. These discs, where the branching occurs, unite it to an adjacent cardiac muscle cell here, and another one shown here that's cut obliquely and leaving the field of view. As one looks without moving the field, scattered within the field, you can see portions of other cardiac muscle cells identified by the position of their intercalated discs, as indicated by the arrow. This is another field of the myocardium illustrating several cardiac myocytes cut in a longitudinal profile. Here's the nucleus of a cardiac muscle cell. Its intercalated disc here. It extends up to the intercalated disc at this location. Note again the myofibrils within the cytoplasm and the central position of the nucleus. Another cardiac myocyte or muscle cell is shown here, the nuclear profile, the accumulation of sarcoplasm containing much glycogen. Its intercalated disc and branching here. Portion of an intercalated disc located here. The nucleus of another cardiac muscle cell here. Its intercalated disc is just beneath the pointer here. And one can see in the background the striations of these myofibrils, which aren't nearly as closely packed as skeletal muscle, and this is why the nuclear profiles remain within the center of the cardiac myocytes. dark nuclei, such as indicated by the pointer scattered within the field, are endothelial cell nuclei, and occasionally one can see remnants of red cells in this cardiac muscle tissue as well, as one would expect, because this is a very, very vascular tissue. So the nuclei of the cardiac myocytes have this character, an open character to them. Occasionally you can see a nucleolus. The very dark, intense staining nuclei belong to endothelial cells making up the capillaries of the cardiac muscle tissue. This is another region of slide number 29, cardiac muscle, to show the myocytes, the cardiac muscle cells, in cross-section or in transverse section, and how they appear when cut at this particular angle. This cell here is a cardiac muscle cell or myocyte cut in cross-section. Note the central position of the nucleus and that rough texture surrounding the nucleus and the cytoplasm are the myofibrils cut in cross-section. This is another cardiac myocyte or muscle cell, centrally positioned nucleus, and once again one can see the myofibrils in cross-section. Another cardiac muscle cell is shown here with the same features, one here and one here. Now, because of the length of these cells, this is another cardiac muscle cell. Note that the nuclear profile is missing. One can see that lake of glycogen and sarcoplasm right here. It's been cut just beyond the nucleus. So profiles of myocytes such as this are beyond the nuclear profile. And if you're fortunate enough to see this lake of glycogen, you know you're fairly close to the nuclear profile of that cardiac myocyte. Once again, the dark, smaller staining nuclei are those of endothelial cells in this very vascular tissue. And if one looks very carefully, one can see a few scattered erythrocytes or red blood cells within this vascular network of this particular tissue, which is to be expected, of course, due to its very vascular nature. Next, examine slides 14 and 22, slides of the aorta, for examples of an elastic or a conducting artery. Look for the three primary layers constituting the wall of this particular vessel. That is the tunica intima, tunica media, and tunica adventitia. Please recall that the three layers, the tunica intima corresponds to the endocardium, the tunica media corresponds to the myocardium of the heart, and finally the tunica adventitia corresponds to the pericardium of the heart. So this entire circulatory system can be broken down into three basic layers and their subcomponents. This is slide number 14, the cross-section of the aorta. The intima is shown right here, the tip of the arrow. From about this point to about here, we can focus this perhaps just a little more crisply, is the tunica media, a substantial layer in this conducting vessel, and the adventitia, this collagenous type of connective tissue and fat lying in this particular region. Now let's go down up at increased magnification, approximately where the tip of the arrow now indicates, and look at the subcomponents of the tunica intima, media, and adventitia. This is that section of the aorta seen at high magnification to show the subcomponents of the three major sub-layers, that is the tunica intima, media, and adventitia. The intima extends from about this point to here. Note that it consists of an endothelial lining. We can see the endothelial cell nucleus and its cytoplasm extending in either direction, another endothelial cell nucleus, another endothelial cell nucleus, and another endothelial cell nucleus. So this is the lining epithelium, the endothelium of the aorta. Immediately underlying the endothelium is a delicate connective tissue that extends from about this point, or where the arrow is indicating. This is referred to as a subendothelial connective tissue. And immediately beneath it lies a large elastic laminae, the scalloped elastic band we're seeing here in section and being traced by the arrow. This is the internal elastic lamina. So those three elements, the endothelial lining, the subendothelial connective tissue, and the internal elastic lamina, constitute the tunica intima. This very large layer subjacent to the external or the internal elastic lamina shown here, that consists primarily of smooth muscle and a little bit of connective tissue. One can still see scattered elastic laminae and fibers within it, but it's characterized by all these smooth muscle nuclei, is the tunica media, a much more robust layer, and you can see at this magnification as we're going through it, has a substantial amount of smooth muscle within its wall, and scattered within it also are elastic fibers and other forms of connective tissue. Now, as we continue to pass through this thick tunica media, finally we run into some external elastic laminae, called the external elastic lamina, that separates the tunica media with its smooth muscle, elastic fibers, reticular and collagen fibers, all produced by the smooth muscle cells in this particular area, from the adventitia, shown here, which consists primarily of collagen fibers and fibroblasts. So these bands here, they're frayed usually, and is the case here, and you can see another one here, is the external lamina. In this case, probably the plural would be the external laminae, because there are several making up a little band here that subdivides or separates the tunica media from the tunica adventitia. Now, while examining the tunica adventitia, one will run across small blood vessels within it, such as indicated by the arrow. These are the vasa vasorum vessels, and they consist of arterioles, venules, and capillaries. And these small vessels, the wall of the artery in the larger vessels is so robust and so thick, it needs its own vascular supply. Thus, vessels come from the external surface, that is, through the adventitia, to supply not only the adventitia, but a outer portion of the media as well. Here we can see a small arteriole coming in to supply, it's coursing towards the media to give its vascular supply. So these vessels, these small vessels, are the nutrient supply for the external wall of the aorta and these larger conducting vessels. Those were the examples that we found right there. We don't need to look any further. See both a venule or a vein and a small artery or an arteriole. So these are the vasa vasorum, and numerous examples of these can be found if one examines the tunica adventitia. This is slide number 22, again of aorta, but this preparation has been stained specifically to demonstrate elastin. What we can see is the intima at this location, the endothelial nuclei shown at the tip of the pointer, and the thin cytoplasmic extent of these endothelial cells between the nuclei. So this is the endothelial lining. This light pink type of material is the subendothelial connective tissue. This huge dense band where the arrow disappears in is the internal elastic lamina. Now, as we course through this thick vessel, what's important to note is the amount of elastin or elastic tissue in addition to the smooth muscle. What's staining now, and very nondescript, that pink type of color. Let me just get it focused here just a little bit more crisply. This material here is actually smooth muscle. The nucleus of a smooth muscle cell is shown here. Nucleus of a smooth muscle cell is shown here. Nucleus of a smooth muscle cell is shown here. And this pinkish material is its cytoplasm or sarcoplasm of those particular smooth muscle cells that spiral around the lumen. What's interesting to note is notice the abundance and continued presence of elastic laminae between smooth muscle bundles as they spiral around the lumen. So the arrow is now illustrating that one couldn't see very well on the H&E preparation these dense, dark-stained elastic laminae as they go through the wall of the media. And there's also some elastic fibers that interconnect these laminae. So in addition to that smooth muscle wall, scattered throughout it and between it are maybe 30 to 40 of these large elastic laminae, that these sheets of elastin that course around the lumen of the aorta. And now we're getting closer to the adventitia. Collagen is now going to take over. There's a few elastic fibers. So the external laminae are perhaps in here. Maybe I can find a better example. We're now ending into the adventitia, where it's primarily a collagenous type of connective tissue, although there will be some scattered elastic fibers within it as well. This is, I move back on a little bit lower power to demonstrate the transition from the tunica media with its smooth muscle and elastic laminae. This is perhaps the external elastic lamina right here, and then finally going in and reaching the tunica adventitia, which consists primarily of collagen spiraling about circumferentially about the aorta. As one can see, I'm running the arrow along its boundary, and it has a different staining properties because it is primarily collagenous in nature. So this is an adventitia, relatively small layer extending where the arrow now indicates. Next, examine slide number 40. This is a neurovascular bundle, actually the carotid sheath, and what it contains is the common carotid artery, as shown here at the arrow. And if we move the slide over, one can see a collapsed jugular vein. Its external wall extends from here to here. Its lumen is collapsed and shown at the tip of the arrow. Let me trace that for you. So it's a fairly large vessel, and here it opens just slightly. If we course around within the tissue, surrounding tissue within this carotid sheath, one can also see smaller caliber blood vessels. And this large solid structure here, this is actually a large nerve, one of the cranial nerves known as the vagus nerves. And you can go down and examine the subcomponents of this particular structure as well. But let's go back up to the vasculature at the present time. As you examine the common carotid artery, it too is one of the conducting arteries, major conducting arteries, and though it is smaller, of course, than the aorta, it will show similar features to the aorta as with regard to its subcomponents. So now let's, because it does so, and you can confirm this by your own observations, let us examine a large vein, that jugular vein, which is shown at the arrow, and look at it at a slight increase in magnification. This is the medium-powered objective of the internal jugular vein in that common carotid sheath we were examining earlier with the scanning objective. One can make out the tunica intima right at the tip of the arrow, primarily the endothelial cell lining. This darker staining is all that the tunica media amounts to. made up of 3 or 4 layers of interwoven smooth muscle cells, and it stains a little bit more intensely because of the muscle cell cytoplasm. And this region from here out to here is a very thick tunica adventitia, which is collagenous type of connective tissue and fibroblasts primarily, and dominates the venous side. This is the internal jugular seen at high magnification to illustrate the subcomponents of its wall. The tunica intima is shown here, and as one can see if looks very carefully, the nuclei of the endothelial cells bulging into the lumen of this particular vessel, so they're shown here. Underlying the endothelial cell layers are very thin and discontinuous, or I guess it is continuous, but very scant, I should say, subendothelial connective tissue layer. This layer here, that's the next layer visible, is the tunica media, and it's made up of smooth muscle cells, maybe 3 or 4 layers that are running at slightly different angles. They don't have that nice tight spiral as the arterial side does. Here you can see individual smooth muscles spiraling out into the connective tissue. The cytoplasmic boundary is shown here on this spindle-shaped cell, and this is its nucleus. Another nucleus of a smooth muscle cell shown here, and it has sort of a spindle shape as well. So the arrow is now covering the extent of the tunica media, so it's just much, much thinner than on the vessel on the arterial side, such as the common carotid. From about this point on to here is all tunica adventitia, and it consists mainly of these collagen bundles that are interwoven but circumferentially arranged around the lumen, and the majority of the nuclei seen in the field are fibroblasts. This is a section through the wall of a muscular or distributing artery. Unfortunately, you don't have a good example in your slide collection to look with, but nonetheless, let's look at some of its feature. It too has a tunica intima shown at the tip of the arrow. The tunica media is almost entirely comprised of smooth muscle and is shown here at the arrow. It extends about this far and dominates the field of view as far as structure goes. This is why it is called a muscular artery, because of this very prominent tunica media made up entirely of smooth muscle. From where the tip of the arrow now resides out to the edge of the tissue, this is all tunica adventitia. Note that the tunica media and tunica adventitia are about the same depth. This is sort of a characteristic feature of the muscular artery. Though difficult to see at this mag, the intima has the 3 subcomponents, as we noted earlier: an endothelial layer, a subendothelial connective tissue, and a very prominent internal elastic lamina. The media is all smooth muscle. Then you have an external elastic lamina separating the tunica adventitia shown here from the tunica media and its heavy smooth muscle now shown at the arrow. So this is an example of a muscular artery, or one of the major distributing arteries, like you're going to find in the majority of vessels in the legs and limbs and what have you, those distributing type arteries. Very quickly, the muscular artery seen at high magnification illustrates some of its features. The endothelial layer is shown right at the tip of the arrow. You can see the nuclei scattered along. Immediately subjacent to it is a very thin, light staining subendothelial connective tissue. Then one can barely make out the large scalloped internal elastic lamina shown here at the tip of the arrow. Because it's an H&E preparation, very, very lightly stained, one can then make out the smooth muscle wall of the tunica media. Then if you look carefully along here, here is the external elastic laminae, frayed once again into 2 or 3 laminae, separating the tunica media shown here from the collagenous connective tissue of the tunica adventitia shown at the top of the field of view. Your next exercise is to return to slide number 5, the esophagus, and examine the connective tissue underlying the epithelial layer lining the esophagus. Remember that non-keratinized stratified squamous epithelium, for blood vessels of various types, and identify various sized vessels. Most of them will be small, but you will see venules, capillaries, and smaller caliber veins in this particular location, just to get the practice of differentiating one from the other. So let's move up at a higher power and just course along this area of the connective tissue and identify several examples of small arteries and veins. An example of a small vein cut along its length. Only the endothelial layer and a very thin intima are present. Immediately beneath it, an arteriole lined by an endothelial lining, so that's mainly the intima, and what's visible is a single wrap of smooth muscle cells. So this is the caliber of an arteriole. An example of a small artery coming into the field. See the intimal lining with the endothelial cells, and what sets it apart without any other distinguishing feature is the muscle layer. It's very donut type shaped, 2 or 3, maybe 4 layers of smooth muscle. It then is cut somewhat obliquely here and then comes back into the field here, and you can see the features once again. This muscle wall, the tunica media, indicates that it is a small artery. A small blood vessel is here, primarily an endothelial layer with a little bit of adventitia, 2 to 3 red cells wide. So this is more the caliber of a venule, and there's some more venules located here of the smaller variety. A small vein shown here. Again, only an endothelial lining with a little bit of supporting intima out about this far. And this is a tiny caliber, or much smaller caliber vein shown at this particular location. Notice the absence in both of these instances of smooth muscle in the wall. A small vein shown here. Note the thinness of its wall, only intima. It is an endothelial lining and a little bit of adventitia, I suppose. This blood vessel cut here a little bit tangentially, recognized immediately because of the smooth muscle wall shown here as being a small artery. Small vein, small vein, small vein. A small vein of larger caliber. One can see the endothelial lining and the supporting adventitia. Its companion vessel here is the artery. Note its tunica media and the abundance of smooth muscle in its wall, maybe 4 layers thick. A small venule coursing here and perhaps going to empty into this vein. Can you identify this vessel as shown in the center of the field of view? Can you identify this fairly large vessel shown in the field of view? Note the thinness of its wall that extends from this point to the lining endothelium. Is that a clue? A good-sized vein cut along its length is shown here within the tissue. So this is still a small vein, but one of larger caliber. The lumen is being crossed by the pointer, and its wall extends from here to here. Here you can see a companion small artery, as indicated by the arrow. And finally, the examples of some capillaries. One is shown at the tip of the arrow. The endothelial nucleus is dark and curved, and you can see a single red cell or erythrocyte within its lumen. Another section through either this or an adjacent capillary is shown here. It's sort of cut lengthwise and curves around such as this, and it has 3 erythrocytes within the lumen, and even though the nuclear profile cannot be seen on this side, one can see the very thin wall of the endothelial cell cytoplasm. Now compare the caliber of the capillary, if I move the field just slightly, with some adjacent small vein, with an adjacent small vein. One is shown here, a venule is shown here, and the small artery is shown here. So one gets an idea of the caliber of these tiny capillaries, which are found almost everywhere in the body.